Well, thank you, Paul. I feel the honour is all mine to be asked to introduce this wonderful symposium. And I hope by the end of it, you'll be convinced that we can now make anything from lignin, including lots of money. But I'm particularly here to give you a perspective on the work of our laureates. And in order to do that, it inevitably means giving you something of a historical perspective. But I want to go just a little bit further back than the 30 years or so that they've been working in the field. 400 million years further back, in fact, to the time depicted in this artist's impression of an early Devonian landscape, when plants were just beginning to come out of the water where they had existed up until then and to colonize the land. And in order to do that, they needed to evolve many adaptations, some of which you can see on this cross-section of an exquisitely preserved fossil of this small little plant, Rhynia. And in this cross-section of a stem, you can see that this plant had begun to develop dense chemical material in its outer layers and also in the center, where it was beginning to develop a vasculature, the beginnings of xylem. And we now know that these dense chemicals included some of the precursors of lignin. Of course, Rhynia could only grow to be about 10 centimeters tall, so further adaptations and evolution was needed before trees could evolve. Those chemicals needed to evolve into polymeric lignin to provide UV protection, to waterproof and rigidify the xylem so that trees could conduct water all the way up from the roots to the top of the trees, and to basically make the wall less penetrable, to make it impenetrable to pathogens. And so I wanted to just illustrate this with a picture of a seasonal tree, this holly from my garden. Without lignin, this tree could really only grow as tall as the moss that you can see here growing on the base. But lignin isn't important just for the wood in the stem. Clearly, that's one of its major requirements in plants. But it's also important, for example, in lots of little specialized tissues in plants. For example, the seed coat within the berries will have lignin in there. And that's really important so that when the birds eat the seeds, it can travel through their digestive system and get dispersed when they excrete it into other environments. So just to look in a little bit more detail about what lignin is actually doing in the plant cell wall, you can see here that the major components of plant cell walls are these yellow microfibrils made up of crystalline and semi-crystalline cellulose, which is linear chains of sugars. But in a very mature wood cell wall, that cellulose and the hemicelluloses become completely encrusted in this phenolic polymer lignin that waterproofs and strengthens and glues the whole structure together. And because of that complicated and gluing structure, lignin is often seen as a problem when we want to get to cellulose sugars in industrial processes. For example, it needs to be removed in pulp and making fibers for other purposes in order to get to nice, clean cellulose fibers. And also in more modern processes where we might want to break down the cellulose into its component sugars, we can do that by pre-treating to get rid of the lignin, then adding enzymes to break down the cellulose into sugars. This is a process known as saccharification. And those sugars can then be fermented by microbes into all kinds of useful chemicals. But the start of this process is to, first of all, get rid of the lignin. And we do that using pre-treatments of harsh chemicals, pressure, and high temperature. And that lignin in the past has often been burned for energy to help fuel those processes. But as you'll hear in many of the talks later today, there are now many more innovative and novel ways that we can use that lignin to give us higher value products. So I want to turn now to giving you a little bit of a perspective on some of the wonderful work by our laureates. So I'm going to really talk in the next couple of slides about what we knew 30 years ago or so when they started in the field and what we now know. So 30 years ago, we had the idea that lignin was made up of three particular types of units, these three alcohols here that give rise to the H, G, and S units in lignin. We knew that enzymes, peroxidases and lactases could connect those units in many different ways with many different types of chemical bonds. But we didn't really have a very good idea of the structure of what that lignin might look like. And as I said to you, when lignin is removed in industrial processes, the harsh chemicals that we use completely destroy the structure. So getting to understand the structure of native lignin in the plant cell wall has really been a huge advance. And you can see over here one of the recent structures that John Ralph has come up with for the structure of native lignin. This is an area that John's been working in for the past 30 years and has evolved through many different models to get to a much better understanding of what lignin really looks like in plant cells. And you can see within this model these different S and G monomers and all these different linkages that bring lignin together in a very complex and irregular polymer. John's work has also increased our understanding of how these units get connected together within the cell wall. And we now know that it's through combinatorial radical coupling reactions. In other words, any chemical that is able to get out into the cell wall and that can make a free radical can potentially be incorporated into lignin. There is no underlying order or sequence of events that is very directly tailored the way there would be in some of the carbohydrate molecules. So really a sea change in our understanding of lignin structure due to the work of John Ralph. Another place where we've really refined our understanding is on the biochemical pathway inside cells that makes those monomers. Now, it's far too early in the morning for you to take in the detail on this slide. I just want you to look at the colors. So 30 years ago, the pathway, actually, we had known for quite a long time, and some of the enzymes on it. But we used to think that this orange pathway was the dominant route through the pathway. And through the work of Bourrian and many others, we now know that the dominant route of that pathway in plant cells is in this blue route, and in grasses, the blue and the pink routes. So we know a lot more about how the process of making these monomers happens, making these units happens within plants. And there have also been many discoveries, several of which Wout contributed to or was the lead on to find new enzymes and even new components that go into lignin. So the major technical advance that really allowed us to understand the pathway for lignin biosynthesis better was the ability to be able to genetically manipulate lignin in plants. And it's a massive honor to be able to say that in front of Professor Mark van Montague, who was really the pioneer of this whole technology. So using genetic manipulation, for example, I'm just going to give you one little example here. We've been able to switch down this CAD enzyme that is involved in converting aldehyde molecules inside the cell into alcohol molecules, and those alcohol molecules are the direct units that normally go into lignin. What we discovered when we switched down the CAD gene was that actually those aldehydes are capable of being incorporated into lignin themselves. So we had a new pathway and new monomers, and in other work, manipulating other genes on the pathway, we've discovered new enzymes. We've also sometimes had other novel results. For example, when we manipulated that CAD gene, and this is an experiment in tobacco, we found that the wood became red because of these new components in the lignin. And Wout, again, was very, very quick to be able to move this kind of technology over from simple model plants into real trees. This is an example of manipulating that CAD gene in poplar, and you can see again this bright red color. So another complete seed change in our understanding of how lignin is made within plant cells. Wout has also been a great champion of the omics. This is where we've been able to take what we know about all of the genes in particular species, and Wout was involved in the development of the full genome sequence of poplar, looking at all of the transcripts within a cell and looking at all of the metabolites within plant cells. And this allows us an unbiased way of getting to discover new genes and new enzymes and new processes on the lignin biosynthetic pathway, and many discoveries and new understanding has been made in that way. John and Wout together have also pioneered new analytical techniques for looking at oligolignals, several of those monolignals connected together within the cell. And John has really been a champion in the use of 2D NMR to better understand lignin structure. And many, many people now use this technique. And what we now know 30 years on is that there's not just 3 monolignals, like I showed you a few slides back, but more like 30 or more different compounds that can be incorporated and have been discovered, have been found in lignin, both in manipulated plants and quite often in native lignins as well. And before I go on to how we might use that lignin, I also just want to pay tribute to the importance of field trials and to Wout's role in championing the importance of field trials. You heard some of that in his speech last night. On the left here, there's a picture of a field trial of these CAD antisense poplars that was carried out in the UK many years ago, that what was involved in. You can see from the red colour in the lignin that the lignin is modified. And you can see from the picture of the plants up at the top, I hope you can see at least, that the modified trees, the trees with modified lignin, grew absolutely indistinguishably from unmodified trees. We couldn't tell the difference by any of the things that we measured until we took off the bark and looked at the lignin. More recently, Wout has manipulated the same gene, but in a much more effective way, using a hairpin construct that really knocks down the activity of the gene much more severely. And in this case, you can see up here in the top photographs, these plants have grown bent away from the direction of the wind, so they're not able to hold themselves upright in quite the same way as an unmodified tree would. And this just shows you how important it is to always take your plants out into the field and really see how plants will behave in a natural environment. This work really shows the parameters that we have in fine-tuning how we would manipulate lignin in plants and in forest trees so that we can use them for industrial benefit. What would we actually use some of those novel lignins to do? So, for example, we could engineer lignin to be more uniform so that when we do deconstruct it and extract it, there's a higher yield of specific chemicals, which would be more useful to industry. We could engineer the bonding in lignin to make it easier to extract in the first place. And John will talk later about his zip lignin, which I think was also mentioned last night. And as I've shown you some examples, we can actually incorporate completely new monomers, which might give us new opportunities to make novel products. So what kind of products could we make? Well, actually, we can't do much worse than going back to nature. You'll hear later today about how we can make products, coatings from lignin that are waterproof and can protect from UV. You'll talk about boards and composites that could be made that are strong and glued together through lignin, and many products where the use of lignin may make the products have a little bit more longevity because it is relatively recalcitrant to break down in the environment. It does, of course, break down, but much more slowly, and sequesters carbon for longer. And many, many other chemicals and applications that I'm sure we don't even conceive of today. So without further ado, I'm going to hand over to the laureates to give you more in-depth stories of some of the pieces of work that they've done. But I also want to thank the organisers very much for inviting me to come and give this introduction.